So our next presenter is Akorede Seriki from the University of Idaho.
Each of us has over 37 trillion cells, but we started life with just one cell. That's one cell divided over and over into the complex multicellular organism that we are. Bacteria, on the other hand, start as one cell and maintain one cell with every division, so they are unicellular. When bacteria divides, it makes a clone, transferring genetic information from the mother to the daughter cell. This continues exponentially, shown in panel A, to form a colony. So a colony is made of clones or cells with the same genetic information. Based on this knowledge, we tend to make inferences about cells in a population by taking the average. But have you ever seen identical twins that were raised similarly but behave differently? Likewise, some cells in a colony can behave different from others. In my research, I use single-cell techniques like microscopy to observe cells individually. With this method, we produce the images in panels B and D of the bacteria I study called Methylarubrum extoquens. Panel B is an image of its cells from the same environment, the same population. But zoom into the part that looks like the map of Africa, where I come from, and we see the cells have different morphology. They all have the same genetic information, but behave differently. We eat food and grow. So does bacteria. For Methylarubrum, that food can be a toxin called formaldehyde. When exposed to lethal concentrations of formaldehyde that should kill all cells in the population because of toxicity, majority die, but few survive and pass on the ability to their offspring. These survivors have different genes, and I aim to understand if and how these genes enable bacteria to tolerate toxin. I do this by observing the cells in the colors red and yellow to distinguish them when the genes are active, shown in panel D. The unique ability of this bacteria to consume lethal toxins can be exploited in biotech industries by using them to get rid of toxins in the environment. More broadly, understanding how cells can be the same genetically, yet act differently, is important in the treatment of diseases. For instance, current treatment against cancer targets majority of cells in the cancer population. But studies have shown that the actual culprits are a minority of cells that are not affected by this treatment because they behave differently. Studying cells one by one can help us to understand how we can identify abnormal cells out of over trillions and target them for treatment. Thank you.
Akorede,
yeah.
You said you're from Africa.
Yes.
What brought you to Idaho?
What?
What brought you to Idaho?
Oh, school, grad school.
Okay,
Research, yeah.
okay. And what is your major?
Biology.
Biology. Mm-hmm. And so when I'm listening to you, you're looking for solutions that might impact both the environment and medicine. Mm-hmm. Is there one field that you're more interested in, or is it just more the study of the bacteria?
Oh, it's more the study of bacteria and basically majorly the environment, like how to use this bacteria to get rid of toxins in the environment. That's like my major focus, but like it has applications in medicine as well.
All right. Mm-hmm. Thank you so much.
Yeah.